All right, let's get started. This is going to be a class on neuropsychopharmacology. Now, of course, that is a really big word. It actually doesn't even fit on one line of a text. But I named this class after some of those anti-drug campaigns of previous decades. So getting started, a little bit about me. My name's Lauren. I am from South Jersey. I moved to Boston. I studied neuroscience, and I now work as a sleep technician. So if you want to ask me any questions about what it's like to watch people sleep all night, I'll take those too. And then a little more about me. I actually first attended a splash in Princeton when I was 15 years old. That's a photo that I decided was a good idea to take at that splash. And it was the first time anyone ever taught me about neuroscience. And now I actually am on the board of the organization that helps run splashes all over the country. So if you have any questions about splashes, ask me those too. So I like to start all of my classes with learning goals. Something that if you walk away and you don't learn anything, hopefully at least you take these away. So the first one that I want to get at is that drugs aren't anything magic. They're not anything supernatural. The only reason that any drug you could ever take works is because you have a neurological system in place that would facilitate it to work. They're kind of like hijackers. The second thing that I really want to get across is that drugs themselves are just chemicals, right? There's nothing good about drugs. There's nothing bad about drugs themselves. They just exist. But where we can get to categorizing good, bad is how drugs are used. So on one end, drug use could be therapeutic. Think like someone taking medical marijuana for their multiple sclerosis. Or drug use could be non-problematic. Think like a recreational smoker who doesn't have any interference with daily life function because of that use. And then on the other hand, drug use can be terribly problematic. So think someone who can hardly get out of bed without smoking, or something of the sort. And the third thing that I really want to get across is that information is good. You might think, I'm never going to do cocaine, why do I need to learn about it? But statistically, these agents are heavily prevalent already in the world that you guys are living in as high school students, and so having a little bit of information could be the difference between your mom coming home and having a few glasses of wine and taking a sleeping pill and it going really poorly, and, instead of you saying, hey mom, you've been drinking, maybe you shouldn't. So, let's talk about the title of the class. And again, if there are any questions throughout this process at all, I'm watching the chat. Also, just turn on your microphone and straight interrupt me. I take that too. So the title of this course, Neuopsychopharmacology. Big word, lot of roots. Let's break it down. So pharma implies that we're studying chemical substances. And then neuro, they act upon the nervous system. And then psycho, what do these agents do? They alter behavior. And behavior is defined as mood, cognition, any actions that you take that are a result of your nervous processes. So there are a few ways that people like to classify different drugs. In general, these aren't necessarily hard and fast classifications because these classifications actually have very little to do with the neuro element. Instead, these classifications are more in tune to how these agents affect behavior. So you might have heard that caffeine is a stimulant, alcohol is a depressant, but the truth is actually a lot more complex than that there are drugs that do multiple things in the nervous system that are both stimulating and depressing. So throughout this class, we'll talk about a number of these agents on here. Alright, so first thing, how do these drugs get in my body, right? So there's a number of different ways that drug administration can occur, and it matters for a lot of different reasons. So taking marijuana for an example, marijuana can be inhaled. It can also be inhaled as vapor, which is an entirely different administration than smoke itself. There are topical administrations of marijuana. These are useful for people with chronic pain, things like that. And then there are many oral ways of administering marijuana, so pot brownies or edibles that you may have heard of. And this matters a whole lot. This matters because it affects how the drug is metabolized in your body and what levels it'll have at any given time in your blood plasma, which can elicit different behavior. Now, for the same way that there are a lot of benefits to differently administered drugs. So, for example, someone who's asthmatic, if you were to take a pill to fix your asthma, that would be a long time without you breathing, but the benefit of an inhaled drug is it acts very quickly and it peaks very fast in the body. So that's one method where that would be useful. But there are also a lot of risks involved. So something that's particularly risky is IV drug use. Intravenous administration of drugs also has that same effect as inhalation, where it happens very quickly. And so for someone who's using heroin or another IV drug, things can get dangerous very fast. So there are benefits, there are risks, and how the drug's administered is really important. So we're going to talk about a really fundamental concept of not only neuroscience, but the scope of this class, and that's neurotransmission. So neurotransmission, breaking it down, all it means is chemical messenger in the nervous system. So what we're looking at here is dopamine, and whenever I say neurotransmitter, I want you to think of a chemical messenger. So how are these messages sent? So you guys might have heard of action potentials, and action potentials can be summarized pretty easily. Let's say that I wanted to turn on the light switch, right? So that signal, I flip the switch, and it goes, through however electricity works, and then the light activates. So similarly, there are electrical impulses in your brain. And so that electrical impulse will travel along the length of one of your nervous cells, and then it'll reach the end. We call that the terminal, and at the terminal, the junction between two neurons is called a synapse. And so at that synapse, a lot of stuff happens, and here's how we get neurotransmission. So once that electrical impulse reaches the end of the cell changes shape, actually. And when it changes shape, calcium is able to enter the cell. And when calcium enters the cell, neurotransmitters can leave the cell. And when neurotransmitters are available in that synapse, in that spot between these neurons, then that's when they're able to bind to the next cell. And that's when they're able to send their message. And that's the basis of how we get all of the action of the drugs that we'll talk about. A few neurotransmitters that we should just look at briefly. So dopamine, when I say dopamine, I want you to think of reward. It's your brain saying, this is good, let's do this again. It propels our species to move forward in life. But I also want you to know that dopamine has an impact on motor function. And so dopaminergic agents are prescribed to treat conditions like schizophrenia or Parkinson's, and some common drugs that act with a lot of dopaminergic activity, cocaine, amphetamines. The next one I want to talk about is serotonin. And when I say serotonin, I want you to think that it's your body's way of saying everything is right, we're okay. So it's satiety, it's am I full enough, am I well slept enough, and it's also mood, am I happy enough, but think kind of an evolutionary sense, but also a very real sense today. And so conditions that we treat with serotonergic drugs are depression, obsessive-compulsive disorder, generalized anxiety disorder, and drugs that act with a lot of serotonergic activity, ecstasy or MDMA, SSRIs. Next, let's talk about GABA. And when I say GABA, I want you to think that it's your brain saying stop. It's your brain saying calm down. And so it's inhibition in the brain. And so conditions that we treat with GABAergic drugs are usually characterized by a lot of overexcitation. So anxiety or seizures or insomnia. And drugs that we would use to treat those conditions, well, alcohol we wouldn't, but benzodiazepines are extremely similar. Things like Valium, Xanax. Let's talk more about the interactions between those neurotransmitters we just talked about, and I'm adding one more. I'm adding norepinephrine, and norepinephrine is really important for your attention. And so the way that these neurotransmitters act, it's not just one or the other doing anything, it's the combinations of them. It's what levels are present in different regions of the brain, and in what regions of the brain do we find these chemical messengers. And so you can see here that attention is mediated not just by dopamine or norepinephrine, but the combination of both of them. The brain is vastly complex. So we can't boil it down to say that one agent does one thing. What's important is the proportions of them to each other and the locations where you find them. And that really gets to my point that everything that we're talking about, it's all chemistry, right? So everything about drugs, how they bind, where they appear in your body, it all has to do with metabolism. It has to do with your body breaking it down in incremental steps. So here's a great anecdotal example of that. This syndrome is called auto-brewery syndrome. And basically what happened is you had a man presenting to the ER appearing to be intoxicated, which was weird because he claimed that he hadn't drank anything. And what it was revealed after, his wife called him a liar and the doctor struggled to believe him for a really long time, is that he actually had a bacteria in his gut that was fermenting bread that he was eating into beer. But yeah, so this is just a perfect example of the fact that alcohol isn't something that appeared out of nowhere. It's something that can be chemically and organically produced within your own body. So let's talk about that hijacking. So drugs work because they look just enough chemically, physically, like these neurotransmitters that you have in your body. So if you see at the top, I have serotonin. That's what serotonin looks like chemically. And you can see that those two rings and the nitrogen-hydrogen structure, that you can find that in all of these drugs that work on serotonin. These drugs only work because your body has a serotonin system, and these drugs are able to hijack that because they look just enough like serotonin. So psilocybin, magic mushrooms, LSD, acid, dimethyltryptamine, commonly brewed in ayahuasca, these are all hallucinogenic drugs, and they all work because of serotonergic activity. The thing that's important about how drugs bind is that there are two different ways they can do it. So there are drug agonists, and these drugs bind to a receptor. I want you to think of receptors and agonists as like locks and keys. So the lock is the receptors that your body, your brain naturally has, and then the key would be the drug, the agent, or a normally occurring neurotransmitter. So when the key is shaped just enough to fit into the lock, then we have drug action and effects. But drugs also can do something really important. They can be antagonistic. So what you see here with this yellow guy is that he looks just enough like the lock to fit in it, but not to elicit any change necessarily. It's like jamming a key in the door, but the door won't open. But what that does is that actually blocks the receptor. And so there's a whole host of other things that drugs can do when that happens. Here's a funny idea. So antagonists would be used, for example, so schizophrenia and Parkinson's are both treated by dopaminergic drugs, but they're treated by opposite dopaminergic drugs. So if you want to think of a condition where someone is, for instance, having too much motor behavior versus a condition where someone's having not enough, then blocking the body's naturally occurring pathway can actually elicit that change. Is that good? I think I'll answer your question better as we go on. So drug action, I just use that term, let's define it. So drug action, we're talking about the specific molecular changes that happen, what happens at the level of the synapse when the drug binds to its receptor or target. So for an example, amphetamine works by reversing the dopamine transporter. The dopamine transporter is important because it takes dopamine that's in the synapse and it brings it back into the cell. But what amphetamine does is it reverses that activity, and instead the dopamine transporter is sending dopamine out of the cell, and it's flooding the receptor. And then we have effects, so what happens because of that? Because of that, we have widespread alterations in physiological or psychological functioning. So with amphetamines, that leads to effects like increased alertness, wakefulness, locomotion, and this can be useful in treating conditions like narcolepsy and ADHD, which are the current indications of amphetamine. But what I want you to really take away from amphetamine is that amphetamine works by flooding that synapse, and when that synapse is flooded, then there's a lot of chemical messages being sent at once. It's kind of like the post office at Christmas time. And so let's talk about tolerance. Now, tolerance has a number of different ways, shapes, or forms that it can take, and going off of that amphetamine example that we're using, what we want to compare is your body at normal state and your body after chronic, long-term use of a drug. So let's say on the left, this normal synapse is what the brain of someone who's never taken an amphetamine looks like. And so the blue guys, they would be dopamine receptors, and the orange star things, those would be dopamine neurotransmitters. And so your body really likes to maintain kind of a balance of things, right? So if your normal function's here, and then with amphetamine, because the synapse is being flooded with dopamine, and suddenly the balance of the system is up here, your body's going to want to do a lot of things to bring it back down. And so what your body does to bring it back down is it down-regulates the receptor site. And so you can see on the right that after this kind of chronic use of amphetamine, because there's so much dopamine in the synapse, that what the body might do is the body might effectively just get rid of some of those receptors, down-regulate. And so the net effect would be similar. That's what your body's trying to maintain. This is called pharmacodynamic tolerance. There's also pharmacokinetic tolerance, and this really just involves the liver and metabolism. And so what's really interesting to note is that people who have been drinking chronically, their liver is actually kind of like a giant sponge for all of the enzymes that your body needs to make to break down these drugs to go through that metabolic pathway that I showed you a vague map of earlier. So the liver is also really trying to pitch into this fight to keep activity at base level. And then there's a third kind of tolerance we'll talk about, and that's psychological tolerance. So this is the idea that, let's say that I have never drank alcohol before and I become intoxicated and my behavior is kind of erratic and I can't walk in a straight line. But let's say that I've been drinking chronically for a very long time and I'm very psychologically used to the behavioral effects of alcohol. I might be able to walk in a straight line just fine because I might have behaviorally adjusted to the effects of that drug. Any questions so far? If you had ceased consuming the drug, then yes, tolerance would decrease. Now, it's different for every agent we could talk about. And every agent that we could talk about, that process of reducing tolerance occupies one or more of these methods that we're talking about now. So with alcohol in particular, there are receptor sites in your brain that would be downregulated. One's responsible for GABA over time, and so your brain would kind of come back to baseline. And even longer, depending on how chronically one's been drinking, the liver can actually recover unless you've kind of passed a point of no return. So that's a process of the liver just ceasing to make as many enzymes. Yeah, so natural tolerance does exist, and natural tolerance does exist for a lot of reasons. So we could talk about the liver, and we could talk about the fact that someone might have genes that encode better forms of enzymes, or better enable the liver to produce enzymes, or for that metabolic process to happen. We could also talk about someone whose brain chemistry kind of normally, in a very simplistic example, instead of initially having 3 of these blue dopamine receptors, they only have 1 to begin with. So there are a lot of variabilities that come into tolerance, and a lot of them are genetic, and a lot of them are epigenetic, or things that have happened throughout your lifetime, and a number of them are also behavioral. So someone who's been around drunk people might be better able to regulate their movement in a straight line than someone who hadn't been. All right, anything else so far? Or I think we'll keep on trucking. All right. Thanks for that question, by the way. So tolerance can lead to this condition that we characterize as dependence. So dependence is basically defined as you are tolerant to the point where if you stop taking the drug, you would enter withdrawal. Kind of a sandwich definition here. And so withdrawal is any number of symptoms that'll appear after one has stopped using an agent. And again, we're talking chronically over a long time. For most drugs, there are examples where this phenomenon happens almost instantaneously. And so to demonstrate the concept, it's kind of that same idea of what comes up must go down. It's that your body is always trying to maintain this equilibrium. And so if the activity of an agent puts your body up here, so to speak, your body's going to do whatever it can to bring it down to here. So it's going to kind of have an opposite reaction. And when that drug is no longer in your body system, all that's left is that opposite reaction. And so that's where we would get withdrawal states. I want to talk for a second about SSRIs. A lot of you might have heard of them. Some of you are probably on them. SSRI stands for Selective Serotonin Reuptake Inhibitor, and we use these to treat conditions like major depressive disorder, generalized anxiety disorder, obsessive compulsive disorder. And so the idea here is that serotonin, that neurotransmitter all about satiety that we talked about, your body wants to take that back up into the cell when it's done being used. If it's not binding to a receptor, your body wants to bring it back into the cell so it can release it again. And what SSRIs do is they actually inhibit your body's ability to take those neurotransmitters back up into your cells. There are a number of drugs that actually work functionally like that, where an addict of one agent or another is basically able to keep using the drug, but because we're administering this special agent, it doesn't have the effect. And so usually what those have to do with is that sense of euphoria. And usually what those secondary drugs do is if someone administers a drug, they're not going to get the euphoric benefit out of it. And so talking more about serotonin, a lot of these drugs that work on serotonin are commonly used as hallucinogens, but I want to talk about medical practicalities of these agents. So psilocybin, magic mushrooms, it is being looked into as a treatment for cluster headaches, of which it's one of only a few discovered. Also addiction. So this is another drug that can improve outcomes and neuroplasticity to respond to addiction as well, which is kind of funny, doesn't it sound like, administering one drug to fix a problem with another. And then also psilocybin trials have been conducted with terminal cancer patients, individuals like that, who are suffering from depression. And then MDMA, or ecstasy, is very, very commonly researched these days and being used for particularly the population of veterans with post-traumatic stress disorder. We'll talk a little bit more about MDMA on the next slide. And then marijuana, it's been well supported that marijuana has unique benefits for chronic pain, especially over opioids, because they work on two different systems, the cannabinoids, the opioids, and one has more benefit than the other. Multiple sclerosis related symptoms, and also nausea, vomiting from chemotherapy. There's actually a special little area in the back of your brain called the I'm forgetting the full word, chemo something trigger zone, and the CTZ. It acts to tell your brain, the drug's about to hit our brain, we need to throw up. And so when people are going under chemotherapy, then that area is activated and they have terrible vomiting. And so the active agent in marijuana will actually bind to that site and prevent that from happening. And then also marijuana has been useful in drug-resistant epilepsy. Ignore my glitchy computer. Charlotte's Web might have been a version of that you've heard of. So talking a little bit more about MDMA. MDMA is actually neurotoxic, right? So I want to talk a little bit about that, and then I want to talk a little bit about why would something that I'm calling neurotoxic be useful as a treatment. So, buzzwords, you might have heard the definitions of these. Neurotoxic, it means bad. Overexcitation, it means bad. Apoptosis, cell death, also means very bad. So, MDMA is a type of amphetamine, actually. And so, that process that we saw where the transporter is reversed, and instead of dopamine being brought back into the synapse, and in this case, serotonin being brought back into the synapse, it's just being dumped into the synapse. And so your brain is kind of overloaded with serotonin. And what you'll see actually in the bottom right, so on the left, let's look at someone who's never used MDMA. And in the middle, that would be someone 2 weeks after they have used it. Is this overexcitation, this too much increase of activity will actually cause some cells to kind of explode and die, which isn't super great. And so you'll see a reduction on the serotonin neurons in the brain. And of course, this phenomenon is recoverable to a degree, and so on the right, you'll see 7 years after ecstasy. So I want you guys to chime in, right? How are you doing reconciling the fact that this drug has these consequences, but it's also, I'm saying, a useful treatment? Does anybody have any idea why that might be? So it's actually more of a philosophical question, so sorry if I tricked you guys. But basically the idea is harm reduction. The idea is risk and reward. So if we're looking at a drug and we're understanding that these are the effects of it, but we're looking at the population that can benefit from treatment, and these are typically what we're looking into are veterans with post-traumatic stress disorder, for which the rate of suicidality is incredibly high. And so we're looking at, all right, so if the long-term prognosis is pretty severe and dire for this population, then it's probably worth this treatment, basically, if it kind of saves their life. And so harm reduction is and balancing of risk and reward is definitely an overt theme for this class. So we'll talk a little bit more about harm reduction efforts in society, but I definitely want you guys to think of this idea of risk, reward, consequence, benefit. Thanks for your contributions, everyone. Alright, so here's a really fun slide. We're talking about reward. Let's talk about reward in the brain. So, cocaine, kind of similar to that amphetamine example, where in a normal synapse you have a normal amount of dopamine, and under the influence of cocaine you have a super high, not normal amount of dopamine available in the synapse to bind to those receptors. And so, reward, I told you to think of it as your brain saying, hey, this is good, do it again. And that system is so important in our brain. It's the reason that you crave cheeseburgers. It's the reason that you, hopefully when you're a little bit older and with protection, have sex. It promotes our species to procreate, to survive, right? And it's long done that. Because, hey, do this again is a really effective way for biological advantage. But the system can be taken advantage of, and cocaine, amphetamine, and really almost all drugs have some effect on this dopaminergic reward system, all drugs of abuse that we're talking about, or drugs of a psychopharmacological variant, where they hijack that body system of, hey, this is good, do this again. And because of that increase in activity, instead of that message, it's, hey, this is great, do this all the time. And so this is how addiction is reinforced positively, not meaning good, but what it is happening. And so, yeah, if you look at this rat here, he will just keep pressing that lever all day, if every time he presses the lever he gets a burst of cocaine. And not because he is any one thing or another that the media might have you believe about people with addiction or people who use drugs, but because that is the system in his brain that is telling him this is a good thing to do again. Little bit of a meme break, also a great time for any questions. And now let's talk about how addiction is reinforced negatively, and not what's bad necessarily, but what's not happening. So let's talk about GABA. And let's talk about alcohol. So I told you GABA, think inhibition in the brain. It's a neurotransmitter that's inhibitory. And alcohol actually works by increasing the effects of GABA. All right, good so far. And that's the action. What effects do we have because of that? Well, alcohol can induce euphoria, sedation, and many functional impairments. And so what this opponent process model dictates, right, is it's going off that same idea of your body trying to maintain homeostasis, your body having these compensatory responses to drugs. So if you're here and the drug brings you up here, your body's going to go this way to bring the net back here, and that's what you're seeing in these graphs right here. So looking at the bottom graph, that's indicating when a drug is in the body, and looking at the top graph, A would be the effects of alcohol, let's say, and B would be what is your body doing to compensate for it? What is your body's tolerance response? And so in the middle column, you'll see that the net effect of that alcohol, it kind of goes up pretty high, and then it goes down, and then there's a little bit of a negative kind of element to it, and that would be something like a hangover. But on the right, what you see that the body is compensating for that alcohol administration. That black B line is way larger, and because of that, the net, what you're looking at in the middle, is that the effects of alcohol are greatly reduced. And these processes are all of those tolerance methods that we talked about. And then what you'll see is after the drug is no longer in the body, there is this great massive downswing. And with alcohol in particular, that's very dangerous, definitely deadly, because withdrawal from alcohol causes overexcitation in the brain. So you can have seizures, not to mention anxiety, tachycardia, tremors, and a whole host of other symptoms. So the opponent process model essentially in a way dictates that the withdrawal of a drug will be opposite to the effects of that drug. And so this is why addiction is negatively reinforced, because if someone were to stop doing this behavior, they would be facing this withdrawal symptom cluster. And so in a lot of cases, it might not necessarily be a heavy influence from that reward center of, hey, let's do this again. It might be, if I were to stop doing this, bad things would happen. And then let's just talk about genetics real quick. I don't want you guys to lose sight of the fact that genes make up our whole biology, and so they of course have a lot of different ways that they can affect drugs and how drugs are metabolized in the body, or what effects they have. And I want to in particular talk about alcohol dehydrogenase 2. So ALDH2 is a gene that encodes an enzyme that breaks down alcohol in the body. And what you'll see is that in certain populations of people, namely Southeast Asian individuals, that instead of having 2 functional copies of this enzyme, a good number of them have only 1 functional copy of this enzyme, right? Because genes encode proteins, and if the protein isn't shaped properly, right? And then a number of them actually have no functional copy. And so what happens when these people drink alcohol is you'll see that their faces will turn red, and there could be a whole host of less inane symptoms happening too. And so this is just a perfect Mendelian pea plant genetics example of how differently drugs can be processed by different bodies. And so this is just a summary of addiction. We talked about how it's reinforced positively, how it's reinforced negatively, and what's important is that addiction is characterized by this craving. It's compulsive. It's not the same as wanting a drug, and people with addiction, they'll often relapse and they'll remit, and that's because of a lot of the things that we talked about. And then addiction, it persists despite any medical, legal, social, financial consequences that anyone might have. And again, that's because of these neurological processes that we just talked about. And this is really important because how many of us are there in this room? Looks like let's say there's 14. That means that at least 2 of us will at some point struggle with addiction. And treatment outcomes aren't great. Only about 10% of those 1 in 7 Americans actually receive specific addiction treatment. Here's a meme for a question break. Any questions so far? So another big overarching concept, I know I'm chock full of them, that I would really like to convey is the idea that biology exists from everywhere that you need a microscope to see up to me, or to improve my self-esteem, something really big like an elephant. And biology, it exists on these different levels, and we can actually examine all of these different levels independently. Am I drinking out of a beaker? Caught. I am absolutely drinking out of a beaker. Thanks for asking. That's my set of drinking glasses in my home. Alright, so back to these biological levels of organization. So with drugs, we talked about what's happening on a molecular level, and that's that action, what's happening with neurotransmitters. Then we talked about the effects, so that's what's happening on a systems level. So in your nervous system, in your metabolic system. And then we talked about behavior, and that's the next broadest level. And so what's happening within an organism and with that organism's behavior. And what we're really going to get into is how drugs manifest in society, how science and society interact. And so now we're looking at a population level. So we're going to talk about opioids, and it's going to be a bit sad, so we'll have a couple of funny joke meme breaks, but I know you guys can make it through it. So nociception, it's a fancy word that means how your body perceives pain in your nervous system. And so opioids, they relieve pain. You might be prescribed them after you get your wisdom teeth out or something of the sort. That effect is called analgesia, removing pain. And so if you're this guy and you decide to hit your hand with a hammer, then the signal of pain will travel from the site that it happened to the spinal cord up to the brain, and then your brain will go, oh, ow, we're in pain. And so that's how nociception happens. But opioids, they're agonists of mu-opioid receptors. So they bind to this receptor to elicit their effects of analgesia. Examples, hydrocodone, oxycodone, morphine, heroin, fentanyl, methadone. Big range of different uses there. And so that nociception model on the left, basically what happens is that you hit your hand with the hammer and the signal, or a pain afferent, that means it's being sent, will be relayed to the spinal cord. And that synapse with the spinal cord, substance P, basically substance pain, it's a neurotransmitter, and that'll be released onto the spinal cord and bind to those receptors, and it'll go up to the brain, and then you'll figure out that you're in pain. But mu-opioid receptors are actually found on those cells that would release the substance pain, the substance P, to the spinal cord, with the bottom cell being the spinal cord in this example. And so what they can actually do, and these are naturally occurring systems like endorphins, gonna blow your mind for a second. Endorphin is a portmanteau of the words endogenous morphine. That's all it means. And so these opioid receptors are found on those pain neurons, those nociceptive neurons, and when opioid neurotransmitters are released like endorphins, then they'll tell that neuron, don't send the pain signal. And so the pain signal never gets sent. It's not that there's any actual injury that's being reduced, it's just your brain is never being told that you're in pain because the opioids and endorphin system, something else is stopping that. And so let's talk about addiction in the scope of this. Let's talk about reward. So if you see on the left, we're highlighting two really important brain systems in the reward pathway. The nucleus accumbens and the ventral tegmental area. Say that 5 times fast. And all of these little blue cyan dots, those are places where opiates can bind in the brain, and you'll see that there's a heavy concentration in this reward center. So if we look on the right, we can take a look at a breakdown of what happens. So starting with 1, we see that morphine is administered. When morphine's administered, it agonizes, it binds to, like a locking key, that mu-opioid receptor. And it tells number 2, it says, don't send the GABA. Now, normally what would be happening in this cell is normally your brain's not releasing dopamine. And normally your brain's not releasing dopamine rewarding you because that GABA cell is saying, hey, stop that, don't send the dopamine signal. But in this case, what the mu-opioid receptor will do is it'll tell that cell to stop sending the GABA, so now no one's telling the dopamine cell not to send the dopamine. And so if you imagine, it's kind of a chain, right? It's kind of like turning off one light switch and then the next one turns on. And that's how that reward signal's released. So that's how they're positively reinforced. Now think back to that opponent process model and let's see how these drugs, opioids, use of them is negatively reinforced in the body. So that opponent process model dictates that the withdrawal will basically be opposite to the effects. And so the effects of opioids, we have pain relief, constipation, respiratory depression, that's incredibly dangerous, and then that euphoria from reward and so on and so forth. So what we'd expect to see out of the withdrawal of these drugs is the complete opposite. So instead of relief of pain, it's pain. Instead of constipation, it's diarrhea. And instead of euphoria, it's dysphoria, and so on and so forth. Meme break, and then things get sadder. I like to poke fun at some of the social ways of prohibiting drug use in society of the latter decades, because I think they were so wholly ineffective, and I think that something like what we're doing now, just learning, is far more important. I think a lot of you guys are leaving this classroom going, well, I'm never doing cocaine, I'm never doing ecstasy, I'm never doing... so my intent's just to inform. Alright, back to the SAD. So here we see a graph demonstrating a number of different use patterns of individuals that were at some point addicted to opioid substances. And so what you see here, if you're looking at the light blue, is that a number of these individuals wind up stopping use of opioids. And then what you'll also see that after someone experienced a yellow, occasionally using the drug, and they transition to blue, that's daily use, that they were never actually really able to go back to using occasionally in any grand sense. And what you'll see that's really sad is a lot of these individuals wind up dead, or they wind up institutionalized, or they wind up on methadone maintenance or something of the sort. And these individuals were young, they were kids, so it doesn't just happen to any one given population. Drug overdoses in America are peaking during the pandemic. It was 64,000 when this graph was made. This is the nicest looking graph I can find. It's now estimated to be about 81,000 for 2020, but statistics are still rolling in. Basically, there were a number of waves of deaths related to opioids. So one of them was when they were popularized as being prescribed as painkillers. And so that was the first wave of how individuals are getting kind of on these substances. And then because a lot of individuals went from that to not having those painkillers, then heroin kind of spiked. And then because heroin supply and demand became more popular, fentanyl, which is an incredibly concentrated opioid, to multiple degrees of magnitude greater than heroin, was being produced and sold, and it's incredibly dangerous. And what's incredibly dangerous about opioid use, it has to do with the administration. So intravenous drugs, I told you, are somewhat more dangerous because they act quicker, they peak higher, faster. The other thing that's really important to note about street drugs is that I'm 23. If I wanted to go buy alcohol, I would walk across the street and I would buy a bottle of Jack Daniels or whatever, and I would know that I bought 750 milliliters, and that 750 mils contains 35% alcohol. Now, if I wanted to go buy heroin, right, I meet up with whoever I'm going to meet up with, and they're not giving me a nice regulated label saying, oh, it's 35% opioids. So it's really dangerous because there's no regulation. But here's where we get into harm reduction. So naloxone is one of the coolest things, I think, ever. So we talked about how opioids are opioid receptor agonists. Now, naloxone's an antagonist. So what naloxone successfully does is it fits in that lock. It's a key that fits in the lock, but it won't turn the door. So you're not getting those effects of pain relief. And instead, what you're doing is it's just kind of sitting there, and it's blocking any of that opioid like heroin or fentanyl from binding. And so naloxone or Narcan is administered as a nasal spray most commonly. Many first responders carry it on their person. And not only will it block those opioid drugs from binding to the receptors, which can again be very dangerous because of central nervous system depression, effects on respiration, breathing, but actually it can also kick out ones that are already bound, which is also pretty cool. So this drug is a life-saving medication. And it's actually available over the counter in every state in the country, but it's sold at $109. So yeah, I could go to CVS right now and I could buy some naloxone or Narcan, which would be especially important if I had someone that I cared about who was using these drugs, or I was using these drugs. But what's interesting is that it doesn't cost $109 to make this drug. It costs between $1 and $10 to make 2 doses of Narcan. And so a lot of people have a lot of thoughts on naloxone, on Narcan. number of places you can get it for free, fire stations, things like that, municipal programs. And a lot of people are angry by that. A lot of people think that they play into the stigma associated with addiction. And I'm not telling you what to think. I'm telling you a lot of reasons why you might lean towards thinking that addiction isn't something that happens to any one person or group of people for any reason. It can happen for a lot of reasons because your brain can naturally reinforce it. But here's where we get to science and society. And so here's some questions, food for thought, chew on them. If you want to provide answers in the chat, go for it. Just going to run through. America has a lot of chronic pain patients. So if these prescription painkillers are so addictive, then how do we treat them? And drugs are illegal in the United States. Everything's Schedule 1. Heroin, for example. And, if someone's addicted, should that be illegal? And harm reduction, should we provide needle exchanges for users so they get clean needles? Rates of hepatitis, HIV also spread greatly when people are using needles that aren't sterile. So some countries have needle exchanges where they'll give you clean ones for dirty ones. And a lot of these people who first responders administer Narcan to, it's not just once, it's maybe 3 times that night, and then maybe they'll relapse and it'll have to be 3 times another night. Should we do that? Is that a waste of resources? Some countries provide supervised injection facilities, places where one can go and inject heroin with medical supervision, nursing staff. They'll test the drug to make sure it is what it says it is and not something in disguise like fentanyl that could be dangerous. And do you think that any of these harm reduction efforts would cause people to want to be addicted more, right? Is that a bad thing? And who funds all this? Where does the money come from? And then who do we even blame for America's opioid epidemic? Is it the doctors who prescribed prescription opioids? Pharmaceutical companies that advertise them? Is it health insurers for not paying for better treatment? Is it the government for allowing it? Is it drug traffickers bringing in fentanyl or heroin? Is it the dealers selling it? Is it the users using it? Is it patients of addiction or a pain condition? And does this even matter? Does any of this even matter? What does matter? So those are some food for thought questions. And just to talk about another intersection of science and society, Portugal actually did something interesting. Oregon's recently done the same thing, where they decriminalized all illicit drugs. So in the United States, Schedule 1 drugs are illicit and perceived to have no medical benefit. And so individuals were then enabled to produce up to a 10-day supply of their drug without committing a crime. So that's not to say that they encouraged it, that's just to say that previously, if you had that quantity on you, it would be illegal. Now it's not. It's just not illegal. It's not necessarily legal, it's just not illegal. And so then what Portugal saw is that cases of infectious diseases like HIV from dirty needles, things like that, they fell dramatically over that decade. And then also overdose deaths decreased heavily in Portugal. And so the idea is that this would have made what is in many governments a legal issue, drug use, it turned into a public health issue. And so it enabled people to have better information, better treatment. So that's just another food for thought. All right, we're going to watch a video, and hopefully you guys enjoy it.
The wood spider is the most accomplished of all web-building species. Recently, scientists gave these tiny creatures a variety of psychoactive drugs to observe their effects on web building. When given a minute dose of LSD, the spider's web took on an unfamiliar, minimalist structure. When given caffeine, the web structure was not affected, but the spider's behavior was. Given THC, the active ingredient in marijuana, the spider didn't build a web. It built a hammock, where it lay all day and watched the caffeine spider go. When given alcohol, the spider built a web, found a mate, and raised over 100 young. But the mate got a restraining order, and now the spider can't go within 100 centimeters of the web. The crack cocaine spider figured building webs was for suckers, waited till the caffeine spider was exhausted, then came up behind it and popped a cap in its ass. Nice web, Mr. Crack Spider. When winter came, the marijuana spider had no place to live. It ended up in the crack web as the crack spider's bitch. For more information on the crack spider's bitch, contact the Canadian Wildlife Service in Ottawa.
Yeah, I just admitted you guys to the waiting room because I figured you'd enjoy the spider video. Thanks for coming, guys.